on confocal fluorescence microscopy. Confocal microscopy can observe thick parts of a specimen as well as in vivo. A comparison of confocal D through F and conventional A through C images across 3 different planes show a much sharper image for confocal imaging. Confocal microscopy, developed and patented by Marvin Minsky in 1957, uses optical imaging to create a virtual slice within the tissue. It provides high-quality images with fine detail and more contrast than conventional microscopy, and the imaging technique also allows for reconstruction of virtual 3D images of the tissue when multiple sections are combined. In combination with fluorescent proteins, the microscope has evolved to allow images and real-time movies of in vitro and even some in vivo cells, which has allowed cell biologists to examine the structure and function of many cellular components and cells as a whole. This is his original confocal design. Throughout the years, it continued to be developed and modified, such as the scanning disc confocal microscope, which spins and has many pinholes. Adding new equipment, confocal laser scanning process, which moves the lasers rather than the specimen. In 1987, the MRC in Oxford is the first to combine confocal microscopy with fluorescence microscopy. This addressed many of the problems associated with earlier forms of the confocal microscope. Programmable Array Microscopes PAM, which incorporates a spatial light modulator. Total Internal Reflectance Fluorescence Microscopy. TIRF, which has the advantage of being able to give images that have very low background fluorescence and virtually no out-of-focus light, reducing the amount of noise in the image and increasing clarity. Things continue to be improved.
The confocal fluorescence microscope is a piece of equipment that differs a lot from the regular compound light microscope undergrads are used to seeing. Of course, we have the well-known parts of the compound light microscope. We have the objective lens, the ocular lens, and the stage. If you look over at the confocal microscope, we can see the same kind of setup, except it has more equipment on the side. But what matters here is how it's internally built. How does a confocal microscope show us highly detailed images of a colorful specimen, and how does it differ from other microscopes? So the confocal fluorescence microscope itself is equipped with parts we're not familiar with yet. It has the pinhole apertures where the laser will transmit its light into. The aperture will limit the light coming through so that there isn't a high amount reflecting that would ruin the overall image. And then we have the filters that are positioned in front of the apertures. The first filter, the excitation filter, will take the light and filter out the wavelengths not necessary for the color the observer wants the specimen to be. The second filter, the emission filter, will block out any other excess light. And then we have the dichroic mirror. This mirror will reflect light of specific wavelengths so that only the desired color of light is transmitted through. Now we have the complete image of the confocal fluorescence microscope at work. You can see a bunch of light bouncing around the parts I just mentioned. Overall, the specimen will reflect the incoming light, which is then sent through the dichroic mirror and to the photomultiplier detector, where the complete image will gather. If we move over to the images of other microscopes once again, we can see that the electron microscope has no similar parts to the other two. Just by looking at it, we know that the most powerful microscope would be the electron, but it still doesn't level with the confocal fluorescence microscope. With the confocal microscope, the observer is able to observe the specimen in real time. Cell functions and processes can be observed, unlike the axis an electron microscope offers. An electron microscope shoots a beam of electrons into a vacuum where the living specimen wouldn't be able to survive. So although it's more powerful and it gives closer images of cells, it still wouldn't provide a look into cellular function.
Going into depth, a confocal microscope consists of a laser, 2 pinholes, a dichroic mirror, objective lens, and a photodetector. Starting with the laser, a beam passes through a light source, being the pinhole. This pinhole increases image resolution and eliminates any out-of-focus light that the specimen bounces off so that the image won't be blurry. Once the laser beam passes through the pinhole, it collides with the dichroic mirror, as shown here. Thus, this mirror reflects short excitation wavelengths from a 90-degree angle onto the specimen right below. As shown here, 90 goes right down to the specimen. The specimen then transmits into an even longer emitted fluorescent wavelength that goes straight into another pinhole, goes right back up, this emission light, and into the detection for imaging. Now, a pinhole allows emissions that originated from the focal plane, being the middle of the specimen, to reach the detector while blocking out-of-focus fluorescence from reaching the detector. Going back to the specimen, once the light hits a specimen, Different color lights may emit from the specimen's molecules known as fluorescence or a type of glow. This increases the molecule's energy and forms excited molecules that give off an emission light. This emission light comes from hitting the focal plane of the specimen emissions from either above or below the focal plane, known as out-of-focus fluorescence, which creates a hazy background that reduces image resolution and contrast. We can see here, specimen is on the focal plane, it goes to the pinhole, and it gives a clear image. Now this is above and below. They don't go to the pinhole, they hit the pinhole and create a hazy background. This is a better understanding. The specimen is on the focus plane, this is above and below, and then these are the results of what happens. So it hits the specimen, it goes right through the pinhole, giving you a clear image. This one is above, doesn't go to the pinhole, but it hits the pinhole, creating a hazy. The same goes for the below. It hits the pinhole and creates a hazy one. Now, once it reaches the detector, which is a highly sensitive piece that collects photons that emit from a specimen, being the emission that bounced off, being the emission light that bounced off. So starting as a light signal, it would then turn into an electrical signal that would take multiple section images, as shown here in the little squares, that the computer will then combine like a puzzle to form one sharp image of the whole specimen, being either 2 or 3D pictures.
Practice of surgical pathology, optical imaging is rapidly evolving. Various non-invasive imaging techniques are being studied and developed as optical imaging platforms are safer, faster, and can be repeated procedures. Optical imaging uses visible light to exploit the properties of photons to produce detailed images of tissues without exposing them to harmful radiation. Such techniques have been developed and are routinely used in real-time in vivo imaging of organs, but ex vivo optical imaging is not yet routinely used, although the modalities for this exist, such as the Confocal Fluorescence microscope. A reason for its scarce use might be because of its limited evidence regarding its feasibility in routine surgical pathology, but a couple of scientists from the University of Texas MD Anderson Cancer Center set out to check its feasibility. They imaged 55 tissues derived from the breast, lung, liver, and kidney. They stained it with acridine orange and viewed it under a confocal fluorescent microscope. The images were then compared to H&E specimen tissues as a control. They acquired the tissues within 5 to 10 minutes and were able to examine and give primary diagnostics within 5 minutes as well. They noticed that the CFM images bear a similar resemblance to the H&E specimen tissues, but the only downside they observed were the small focal areas, and the CFM showed a lack of sharpness, but it was noted that it only occurred in a few specimens. The CFM images clearly showed the utility of the non-invasive imaging, its ability to reproduce recognizable architecture features in the samples, and its feasibility for rapid examination, as well as having advantages over frozen section analysis, which is a common practice in surgical pathology, as freezing tissues for later examination can take up to 30 minutes to prepare, cause artifacts in the tissue, and has a lot of tissue loss compared to CFM.